Hi, everybody. Thank you so much. It's been such a wonderful morning listening to a lot of really fun stories and great science as well. My name is Arun Sethuraman. I'm an associate professor of biology, computational biology at San Diego State University. I'm going to tell you a little bit about some work that we've done recently in developing new computational methods to estimate the timing of auto and allopolyploidy events. But before I start, I got inspired by Roland and Lorena, and I put in some of these cool pictures that I found on my computer of all the cool activism and important stuff that we do apart from the science in my lab. My lab's very diverse. We're a bunch of really, queer identifying or queer ally folks. We do a lot of activism both in and around San Diego, including walking in the San Diego Pride Parade. If you are in San Diego or happen to be here, I encourage you to come say hi on July 20th. My whole lab will be walking with me. You can learn more about some of the stuff that we do in my lab at that QR code. Okay. Um, why is this project that we're doing important? The whole idea boils down to the fact that whole genome duplication events. So this is when you have instantaneous genome duplications that result in instant speciation. It seems to be pretty much very, very common and cyclic across plantae. So here's a nice adopted figure from Clark and Donahue where they looked across plants. So here's eudicots, monocots. And what they determined was that you've got these sort of ancestral duplication events that have happened pretty much throughout the history of all plants. Somehow plants seem to be able to do a lot much more stable conditions with whole genome duplications than animals, but that's a discussion for another point. But what's interesting here is that a majority of these duplication events are of unknown origins. And what I mean by that is that the two most common types of duplications that are present across all plantae are either autopolyploidy events, where you have one single whole genome duplication event that then causes genomes to sort of double within the cell, and then you have instant speciation. Or you have what's called allopolyploidy events, where you have whole genome duplications that have happened somewhere in the past, and then the two species just diverge. And then at some point in evolutionary time, they continue to exchange genes, and now you suddenly have a triploid allopolyploid species. And we still don't know a majority of these blue circles out here if they're auto or allopolyploids. And so why is this important? Whole genome duplication has resulted in novelty of genes, genetic material. But then again, a majority of these whole genome duplication events are also under very strong selection, which results in huge purging of these gene or genetic material right after the whole genome duplication events. So then our lab is sort of really interested in, how do these auto and allopolyploidy events, specifically the mode of speciation in these cases, what is the evolutionary consequence of these, especially when we look at analyzing large-scale whole genomes in angiosperms in current day time scales. And so one common way that folks actually use to distinguish between auto and allopolyploidy events is by using something called a KS histogram. And this KS is the same that we commonly know as KAKS. The KS here is just the average number of synonymous substitutions between paralogs. And so if you have a whole genome duplication event, let's say in an autopolyploid, and you have genomes that are diverging separately, you just literally take the paralogs in the same species, and then you count up the total number of synonymous mutations between them, and then you average that across all sites, and that would be your the KS distribution. And then you make a histogram of that, and as it turns out, the value of this KS distribution, as you go backwards in time, you'll see several of these plots that I'll show later on. The KS is on the x-axis here, and that happens to be a proxy for evolutionary time. And then the distribution, the histogram, is just the number of paralogs that have accumulated between divergent species. And so our hypothesis was that if we can build an evolutionary computational platform that can then simulate these KS distributions for different types of speciation events, in this case, allo and autopolyploidy events, and then generate the KS plots as output, and then use that to build a discrimination model, which then takes these KS plots as input and classifies these as either auto or allopolyploid, and then also measure the accuracy of these. So we went ahead and did that. We built a method called SPECS, speciation under KS. And so this is just the overall schematic of how this simulation actually runs. We have separate kinds of simulations that we do for autopolyploid events. and then we have separate ones for allopolyploid events. And we hypothesized that the inherent shapes of the KS histograms would be very, very different, as you can kind of see by the schematic here. And so the method itself, broadly, it has several different steps to it. We generate the species tree. We then generate individual gene trees within the species tree. And then we simulate gene birth and death processes along each one of these branches. And then we shed some of these onologues or paralogs, gene evolution. We compute the KS values, and then we generate the histograms, and then we compute results from that. So here are some results. So our first hypothesis was, hey, guess what? Are the auto and allopolyploids actually completely different in the shapes that they generate? They are, as we can see from this plot right here. Like I said, the x-axis here is proportionate to time in millions of years ago, and the y-axis here gives you just the total number of paralogs between the auto and allopolyploids. As you can see, the allopolyploid KS histogram has a much more sort of blunt shape to it, while the autopolyploid one has a much more typical sort of exponential, kind of goes up and then feeds down exponentially. So yes, indeed, they have very different shapes, which means we can actually utilize these to fit these to existing species. So for instance, here I fit these to coffee, corn, and poplars. And you can see that the simulations are able to actually recapitulate the actual shapes of the observed histograms from whole genomic data from across a variety of different species. We've now done that for hundreds of species as well. We can also build a discrimination model, which we did, and you actually get the output, and you can say, hey, guess what? What we did was we built a training set. We built thousands of simulations, 2/3 of which we used to train and 1/3 of it to test. And we see almost 100% accuracy in our estimations, which is really nice. We can actually split the simulated models as either being true autopolyploids or true allopolyploids. And then we also did this across a variety of different species. We built a logistical regression model to do this. And then we did this for over 200 plus whole genome duplication data sets from the 1KP, which is whole transcriptome data from across angiosperms. And we were able to accurately classify these as either containing auto or allopolyploid events. So which is really fun. I'm super stoked about this. Tamsin, my graduate student, has now put together a really nice platform to be able to share this. All the code is on her GitHub page. We invite folks that might be interested to try it out. You can scan the QR code here if you want to access this. And we also have a preprint on this, which we just got posted a couple of weeks ago. And I invite you folks to take a look at this as well. You can also scan the QR code here to learn more. With that, I will... that's all I had. I'll take some questions.
Thank you, Arun, for your presentation. Do you have any question from the audience? I have one. This is very interesting because you are able to see how these things have happened millions of years in the past.
Yep.
My question with evolution analysis on this is, how would you apply it to today's problems, perhaps in the crops fields, commodities, or can you explain a little bit maybe for all of us who are not experts about it?
Yeah, sure. Happy to, actually. So, crops have pretty much all crops have undergone domestication, which is a evolutionary process, which has accelerated either the rate of evolution at specific regions of the genome, which might be much more involved in agronomically important traits. So the next step that we're actually doing, now that we know that we can simulate these sort of large-scale whole genomic data under these specific evolutionary models, is our goal is to throw in selection into it. So then we can actually predict to see what's called genomic prediction, which is can we actually simulate very strong selection across, say, agronomically important genes across plant genomes, and then use that information as to feedback and predict which genes would be under very strong selection or the rates of evolution at some of those genes in the future. So that's kind of our next plan, if you might. Plant genomes are a hot mess. They've got transposable element content just going crazy. They've got repeat content that are just everywhere. They're large genomes. And so we're increasingly learning a lot much more about the complexity of these genomes. And I think as computational biologists, our goal is to sort of simulate that, or be able to simulate that to some accurate level, so then we can use that for genomic prediction. Yeah. I hope that answers your question. So.
Yeah, pretty much. Any question, brother, from the audience?
I have one on plants. I know nothing about plants. I'm not even a good gardener. My partner has to do all of that. For genome annotations, I mean, I work in a cancer center, so I'm looking mostly at mammalian orthologs of proteins I'm interested in. And, quite often the exon intron boundaries are not correct. Like, you see BRCA1 should have all of these different regions. And in some, in the panda sequence is missing some important chunk or something. Is that true in plants that, exon intron boundaries are hard and actually getting the protein sequences out is?
We do, yeah. Plant genomes are, incredibly crazy, especially when you have these sort of sudden bursts of transposable element activity. Things just move around like crazy. So you got whole scale sort of, genome duplications resulting in repeat content, but you also have sort of small scale duplications that are also rife across the genome. So yes, they are messy. And I think plant pan genomes are sort of just getting out there. You see many more mammalian pan genomes or microbial pan genomes, but then I think plants they're increasingly difficult because of just overall high diversity, high heterozygosity, high effector size, that kind of stuff. So yeah, they're kind of messy. I'm not a plant biologist myself either, but I'm learning. So.
Cool.